Thanks for joining us on the Cultured Meat and Future Food Show. We're excited to announce the New Harvest Fellowship Series. Established in 2004, New Harvest is the nonprofit research institution building the field of cellular agriculture. We're partnering with New Harvest for a multi-part series on the Cultured Meat and Future Food Show. We've had conversations with members of New Harvest leadership previously on this show. Please see our episodes with Executive Director Isha Datar. We're excited to have Sam Peabody as the guest for the second episode in the New Harvest Fellowship series. Samuel S. Peabody IV is a first-year PhD student of animal science at Texas Tech University. His area of research includes the study of microbiology and food safety. His current studies are examining the safety aspects of cell-cultured meat, poultry, and seafood. When he's not busy working in the lab, Sam spends his free time gaming and role-playing with friends, lifting weights, hiking, and biking. Sam has always had an interest in cooking and baking, and is especially proud of his Tex-Mex recipes. This was a great and super informative episode. Let's jump right in. Sam, I'm excited to welcome you to the Cultured Meat and Future Food Show.
Thanks for inviting me on, Alex. I appreciate being here. Excited.
Sam, tell us a little bit about your background and when you actually first heard about cultured meat.
So when I first heard about it there was a press release. It might have been one of the ones that was done for Dr. Mark Post's lab and the research and the products that they had developed. At the time, I was still working on my undergraduate in food science, and at that time, I wasn't really sure where I was, what I was wanting to do as a career, but I did know that I liked science, and food science had been a fit for me for a while. So I kept working on my undergraduate degree in food science. And during the last year in that program, I had the opportunity to do some work in a laboratory on Texas Tech campus. It's a laboratory that's part of the International Center of Food Industry Excellence. And while working there, I became acquainted with handling and preparing different kinds of media and agar for growing bacteria. I became familiar with a lot of the intricacies of food microbiology. And I worked there long enough that after a while I had the opportunity to apply for a master's program in food science at the same university under the professor who had hired me as a assistant in the lab for undergraduate.
Cool. And which university was this?
Texas Tech University.
Okay, Texas Tech. And so what year was this that you first heard about that press release?
Oh gosh, that would have been years ago. I'm not going to give you a specific date because I honestly cannot recall. It's been so long, it seems. Maybe 2012, 2013, somewhere in that ballpark.
Oh, interesting. Okay, so right around the time actually that Mark Post was developing I guess making the announcements for his unveiling.
Yeah. It was one of those press releases. That's when I first heard about it, and it was something that just was on the back burner in my head for a while. And it wasn't until I was in my master's and I was starting to see how I could really be a part of the research community and really learn how to develop and get involved in the technology and not just be somebody who has a bachelor's degree in it, but a real scientist working on answering questions and curiosities and inventing solutions for feeding the world. And it was at that point when I remembered that press release and I looked up more information about it. And it led to me discovering New Harvest and a bunch of other things. And since then, it's just been snowballing to the point where I'm here today.
Your undergrad was in food science. When you kind of transitioned to that lab setting, was that a pretty unique thing for you to kind of have this food science experience and be in the lab working on bacteria?
Well, I wouldn't call it unique. There are many schools across the nation where students involved in food science can get involved in a microbiology lab. What's unique about the laboratory at the International Center of Food Industry Excellence is it's actually a series of laboratories, and they are deeply rooted in the local culture and community. which has a lot to do with animal production and meat science. And so there's a deep connection to that, and so we get a lot of exposure to meat science and food safety as applied to meat and poultry products. And so that's the focus in our laboratories. And our laboratories, there's three main laboratories that I've worked in. One is a conventional microbiology lab. We grow agar, we raise bacteria on agar, we count agar plates, and we look at how many colonies of bacteria there are on each plate. And we just do the basic run of the mill, ordinary things that you'd experience in a microbiology lab. We have some really cool tools in that lab as well. We have some immunomagnetic separators, where you take a magnetic bead, you coat it with we'll say with something that's only attaches to a certain type of bacteria, and you can coat these little magnetic beads and they'll only stick to those specific types of bacteria, and so they get coated in them, and then you can use a magnetic field to separate out those bacteria from the rest of your solution. It's a really cool thing.
Wow. Okay.
Other things we have in that lab. We have just about every kind of agar you can think of for food safety in that lab. And over the years, I've gotten a chance to make quite a few different varieties. The other labs that we have, one is devoted to DNA, RNA, qPCR, all those different types of molecular methods that are used. We've done a lot of work developing and improving rapid methods, going back and forth between those two labs. And the most novel experience among these labs, I said there were three. The third lab is the Pathogen Lab. And that's a lab where we bring in equipment that is identical to real equipment used in the meat and poultry industry for processing. And we're able to incorporate actual live pathogens into the processes and steps to evaluate the performance of certain interventions and certain strategies, control strategies, with the actual foodborne pathogens. So when I say intervention, what I'm talking about is a step, a process that reduces the concentration of microorganisms in a food. So, for instance, that could be smoking a turkey at 400 degrees for 20, 30 minutes, or that could be sous vide cooking packages of hot dogs to mitigate the risk of Listeria monocytogenes contamination. An intervention is usually a thermal process, but there can be chemical or even biological approaches. And interventions happen throughout the process of getting food produced from farm to fork. And so we evaluate the performance in that lab.
Wow, cool. Okay, so would you say that this type of lab structure is something that Texas Tech specializes in, or is this fairly common in terms of like food safety microbiology?
I would say the way we have it arrayed and organized is unlike most of their schools out there. The amount of real estate that we have is substantial. We're talking rows and rows and rows and rows of benches. We can have 1,000 samples on benches for processing and have a team of 10, 11, 15 people working together to process all the samples and get them loaded up. So we can do very large scale testing. There's plenty of other schools that can do it just as well, but I don't imagine there are many schools that can do it better.
So I recently did a very, very basic like food safety course, right? And even from that very basic food safety course, I'll walk into a restaurant now and I'll notice like, wow, actually this is not to code, that is not to code, and then I'll be a little bit left out. So
Serve safe.
well, yeah, yeah, exactly. Yeah. So, and you'd be surprised, like some of the basic stuffs, like 6 inches off the ground, and all this kind of stuff is not even implemented in some of these restaurants that I was going to in downtown Berkeley. Which I guess in some ways not to be surprised, but for you being kind of really an expert when it comes to bacteria and especially in relation to food safety, do you ever walk into a restaurant and think like, okay, this is definitely not good?
Yes. There are specific experiences that are an immediate red flag for me. The biggest one for me is there was this restaurant, and I won't say where it is located. And as soon as I walked in, I could smell this musty smell, and it's the smell of rancid oil. Now, There's a couple reasons a smell like that can occur. If it's a very old restaurant, it probably just needs a real deep cleaning. But if it's a relatively new restaurant, like let's say less than 20 years old, that is just a huge warning flag, because it means that even if they're cleaning their countertops and the areas that are regularly used, it means that there are some other areas that they haven't given attention to in a long time. For example, every every grill, every grill top, every cooking surface has these large intake vents above them that pull up all the moisture and steam and oil that gets evaporated during the cooking process. And that oil is important because it can actually build up in the ventilation ducts. And so in some restaurants where this oil builds up on the walls, on surfaces, it collects and it gets old and it creates this musty, rancid smell. Another thing that can cause that are pests, vermin. Roaches have this oily smell to them that's distinct. And so when I go into a restaurant, if I can smell that, that's for me that's a red flag. That's saying, okay, something in this restaurant has not been taken care of in a long while. And if that's not being taken care of, what else aren't they not taking care of? And so if I don't notice that right away, okay, I'm reassured. The next thing I usually look for in a restaurant is I look to see if there's anything on the floor in the kitchen. If I can see the kitchen and I can see the floor, that's usually a good indication to me whether or not the team that's working there are they cleaning as they go. If it's during rush hour and I'm coming in the last 45 minutes of it, I won't think about it too much, because obviously when you're working and you're going full speed, you may not have time to clean then. But if it's say at 3 in the afternoon and I go to a burger joint and I see a restaurant that has obviously got something that's been sitting on the floor for a while, just some scraps of lettuce from preparing a salad, or who knows what. If I see that, my impression is, okay, somebody's not taking the time to clean as they go. What else is not happening in here? And I wouldn't say that that's a reason to not go to a place, because any restaurant can have these kinds of violations at any time. It really comes down to the management and how consistent they are in addressing their problems. And that's why it's great to just look at like local news reports and see where there are health reports coming out, and if the restaurants that do get marked or written up, if they actually do correct those actions, and you can see which ones are repeat offenders.
Wow, yeah. I'll definitely be checking that out, especially now that restaurants are starting to slowly open up from COVID-19. So, transitioning to what you're currently working on now through the new Harvest Fellowship, what are you working on and why is it important for cell cultured meat?
Okay, so right now what I'm working on is looking at the food safety aspects of cell cultured meat, because so far there is limited discussion on how safe these products. And when I'm talking safety, I'm focusing in on the microbiological aspects of it. When we talk about food safety, that's 8 out of 10 of what we're talking about. Sometimes we're looking at chemical or physical hazards, but that's rare. 7 out of 10, 8 out of 10, whatever number you want to use. Most of the time, we're looking at the microbiological aspects of food safety. And what my work is doing is looking at what are the ways in which we can control and minimize bacterial pathways into these foods, and how can we control them in such a way that the establishments that are producing these products are still able to deliver a product that is satisfactory and appealing to consumers. And because there is no information out, public information out on that, we're starting with just the very basics of what can be done. We're building growth models, and we're going to use those growth models as a way of saying, okay, once we see how bacteria grow in this situation, then we can start to see what we can do about it. So going to very basic things, looking at, I put bacteria in this growth media that's used for culturing bovine cells. I put bacteria in there and I see how fast does it grow? When does it become apparent in that situation? And how quickly would we need to be able to respond to that? How quickly would we be able to detect that? Those are the kinds of things we're starting with. Later on, we'll want to we'll want to talk about, well, if we do have this kind of problem, what can we do to stop it? Because right now we're talking about in the growth stage, but then there's also on the other end of it when we're starting to package, process, and put this product on the store shelves. How long is it going to last on those store shelves before bacteria that do get in manage to spoil the product? What kind of risks will there be? Like the processing facility that produces cell culture meat. Like let's say you have the plant that wants to specialize in cell cultured seafood, for example, and they're wanting to produce some kind of cell based sushi or cell cultured sushi. And that's a product that's served raw. How long can that stay on the shelf and still be regarded as safe? If it's going to be incorporated into a sushi roll, then it's going to be exposed to other bacterial organisms. My first thoughts are rice, Bacillus cereus is commonly implicated in rice. Could that get a boost or a benefit from that? It's under refrigeration, so sushi is refrigerated, so Listeria monocytogenes, which is something I studied during my master's, that bacteria can grow at refrigerated temperatures. Would that be a risk in cell cultured seafood, cell cultured sushi?
So theoretically, it could actually be better or worse in terms of the amount of bacteria that is collected when you're comparing it to like traditional fish. Or is it a totally different thing and can't really make a comparison?
Well, we will make comparisons, but we'll let the evidence speak for itself. As far as I can tell, the culture media are rich in sugar, and some of the supplements that are used supply a abundant amount of protein and or amino acids. And so it's a great environment for cultivating bacteria. And I would not be surprised if all kinds of bacteria can turn up. And so what we've really got to look at is how well will these establishments maintain hygiene at their production plants. If they do a great job, a stellar job of maintaining hygiene, there is an opportunity for the level of contamination and the risk of contamination to be lower because they're not harvesting from live animals. Because live animal harvesting involves not only the processing and handling and moving the inside and the outside of an animal. And if you've ever been on a farm, you encounter lots of poop. And so there's poop on the animal, and it gets washed, the hides get washed off, they get cleaned before they're ever on the kill floor. Nevertheless, it's possible for bacteria that were abundant on the animal to then be transferred later on in the path. And the goal with cell cultured meat and poultry and seafood is to do it straight from cells. And so there's no viable way of cultivating these cells that would tolerate the presence of bacteria yet. I don't expect that they can cultivate these cells with any amount of bacteria presence. I wouldn't expect it. I know that sometimes they get mycobacteria in these cell cultures, and that sometimes ruins the student's project because it turns out that it was in their original samples and they have to start all over because the mycobacteria are pervasive and really sneaky like that. But yeah, it'll be interesting to see how well these cell culture meat producers maintain sanitary conditions. I believe they have the potential to be very clean and to be very effective at controlling bacteria in their foods. But we got to see it first.
I've heard people at conferences within the industry say like, oh, well, there won't be any bacteria because you are culturing it in this sterile environment. And they even go as far as to say like 0% bacteria. But I think that any scientists using 0%, especially when it comes to bacteria, is totally wrong. There's always going to be a little bit, or is that not the case? It could be 0%.
I'm reminded of the challenges that microchip manufacturers have to go to. It's a funny tangent, but bear with me on it. So the microchips that are in our computers that make up our processors and all the other little bits and bobs of a computer, these chips have to be constructed in a very intricate and detailed way. And they're manufactured in these clean rooms, these huge factory floors. And you see the people that are going in there, and they've got N95 masks, and they've got hairnets, and they've got these lab coats. They may even have special booties on, and they have certain processes for entering and exiting.
This reminds me of those Intel ads with the guys that are pretty much in hazmat suits.
Exactly. Yeah, they have these kinds of clean rooms. And it might surprise you, but a lot of A lot of food processors have a similar dress that they wear. Not to the same degree of protection, but they do wear substantial coverings over themselves. They wear gloves, they wear hairnets, they may wear masks. It depends where you are in the world. Different countries have different regulations. Nevertheless, all countries have several layers of protection that keep people from unintentionally contaminating food because people themselves carry bacteria. A good organism to look at, and one that we are looking at, one I was working with before COVID-19 popped up, was Staphylococcus aureus. Staphylococcus aureus, well, you know it as the cause of staph infections. There's a version of it out there called MRSA, MRSA, methicillin resistant Staphylococcus aureus, that scares people because it's resistant to certain types of antibiotics. And the thing about Staphylococcus aureus is that that bacteria is colonized on a significant portion of the human population. I've seen numbers around 40% of all people carry it. And so it's nothing to be alarmed about. Obviously, it's not making people sick all the time, but it has the potential to when it grows in food and it produces a certain toxin. And so naturally, if you're going to have people working at your processing plant, if you're bringing in people, you're bringing in what's on people. And we can't always see or tell what falls off of people while they're working in that kind of space. So there's as long as there's a human element, there's going to be a microbial element.
I think of like brewing beer, there's these big tanks and it's very much separated. Would you say that there's still opportunity for bacteria to get in, even in that kind of image?
Well, I'm glad you brought up that analogy because I had the chance for a time to volunteer some of my time for a brewery. And the nice thing about brewing beer is yeast are very good at what they do. They respond very well when you treat them right and give them the right amount of love and care and attention. Anybody who's ever brewed their own beer has a certain affectionate feel towards the yeast that are doing the work, making delicious beer for them. And as it happens, yeast have had this unique quality to them where they're able to fend off their competitors. Most microorganisms have one or two or three tools that they can use to sort of say, hey, get out of here, this is my space. Some bacteria, like lactic acid bacteria, produce what are called bacteriocins, and they're essentially a sort of antimicrobial. In fact, penicillin, classic example of an antibiotic, it was discovered why? Because a form of mold was producing it on petri plates. And so yeast are able to protect the wort that they're incorporated into because there's a high concentration that's added. They start growing in there. If there's anything else that gets in, the yeast are able to kick it out. Will we see a proliferation stage where large vats of mammalian cells are cultivated? I can't imagine it'll be like a brewery. What I recall, and this is going back to some old press releases, I recall in one image there was a storage unit and it had trays and trays and trays and trays stacked on top of each other. And each tray had so many wells in which cell cultures were being produced. And I don't know if that was during their differentiation or proliferation or some combination there too. Nevertheless, I see it as an example of what can be done. If instead of, imagine if you will, instead of a 96 well plate, imagine a larger container, either designed for two-dimensional or three-dimensional growth. I won't conjecture. That's up to the biotechnologists that are working on solving 2D and 3D problems to figure out. But I imagine some version of one of those approaches being scalable, responding to what method works best, having on one side an input. It could be a hose that then breaks up into a bunch of little hoses with backflow preventers that then feed a bunch of different cases all at once so that they can be collected later for forming a larger product. Or it could be something more elegant where it's like a vacuum bag that contains enough of the cells at the start, and once it gets going and going and going, it sort of inflates with cells. I could imagine something like that being a possibility. I can't imagine it'll be exactly like a brewery tank, although if somebody figures out how to do it that way, then great. I don't know that it will look like that. Can that kind of environment be handled in such a way as to prevent any kind of contamination? I think so. I think there are techniques that will need to be developed to overcome the challenges of that. But I really see having a modular unit of some predetermined volume, say a liter, that has all the surfacing or three-dimensional surfacing that's necessary to cultivate these cells. And I imagine a large number of those being stacked or arrayed together that can then be, at some later point in the process, combined or integrated in some way that allows them to then produce a single unit of product, and for there to be a sort of cycle, a life cycle of these units so that they go back and forth. That's how we've seen it done and work. And if a small amount of bacteria were to get into a proliferation tank, like what you said, 100-gallon tank, 1,000-gallon tank, a small amount of bacteria can very rapidly multiply, and it may not be detectable until it's too late and the whole batch needs to be thrown out.
And that could be very costly.
Yes, and that's why I see being able to produce these cells, proliferate these cells in parallel is great, because if you can limit the amount that you lose at a time, you can keep from having a huge loss. And it's an optimization process problem too, because if you work too small on small scale, then you're having to clean and manage and maintain too many pieces of equipment, and that costs you too much time and money. So there's a precarious balance, and right now we don't know if there is a place in between where we can truly balance and make everything happen or not.
So what would, at a very high level, what would be one of the methods of detecting whether bacteria has breached, like a small, let's say like a 1 liter or a smaller vessel, for example?
Well, the options available to cell cultured meat, poultry, and seafood establishments are the same that would be available to any other producer that's out there. There are a variety of different methods. There are rapid methods which use DNA or RNA extraction from the product, and you can use those to quickly identify whether or not there is something foreign present by just having a template in the master mix that binds to the bacterial DNA or bacterial RNA, whatever it is you're looking for. And you can approach that in a qPCR way and get a very strong signal very quickly. There are some limits to detection, though. With those methods, I think the limit is somewhere around 2 logs of bacteria. And when I say log of bacteria, I'm talking about taking the log base 10 of a number, and that's your result. So if I was saying log base 10 of 10, that would be 1. Log base 10 of 100 would be 2. Log of 10,000 would be 4. And so there's a limit of about 2 logs, I believe. And so you can't detect the bacteria if they are under 2 colonies per milliliter. There are other methods out there that are a little more sensitive. What comes to my mind are just traditional plate counts, and these can be done in a variety of ways. There's also a technique that I try to avoid using because it's very time consuming, called most probable number. That can be sensitive enough to get you counts even when the concentration is below 2 logs per mil. However, in those latter 2 cases, those methods are not rapid. Those methods take at least a day, if not several days, to obtain results. It really depends on what you're trying to look for. What we would need in this kind of environment is we would need some way of being able to see within hours whether or not there's contamination, so that we can throw out what's no longer salvageable and save what is. Assuming, of course, there is anything that can be saved. We have no intervention strategies as yet for controlling bacteria in these environments, aside from using large amounts of antibiotics.
So this might be a good time to transition to one of the audience questions. So we actually have a couple audience questions that have come in. And one of them is, will it be free from antibiotics? So let's say once we do have a product, will that product be free of antibiotics?
Well, it will be free of antibiotics for the consumer. I can guarantee you that. There won't be any antibiotics in the product by the time it reaches the store shelf. Or to put another way, there better not be, or the USDA is going to notify the establishment of what they found and ask what's going on.
Well, actually, so maybe this is a good opportunity to demystify a couple of, I guess, potentially misconceptions in the industry. Like people believe that, it's okay to have antibiotics in food, but in reality, if antibiotics are ever used during livestock production, they have to wait a pretty tremendous kind of factor to make sure that it's out of the system of the animal before they're sent to slaughter. Is that right?
Yes, I'm not going to go too much into detail of while the animal is alive. Um, I just don't have as much familiarity with that side as I would like to. But I can tell you that Food Safety Inspection Service, FSIS, they will take a swab of the kidneys of cattle after harvesting, and those kidney swabs that they take, they send those out to a laboratory where they do analysis and they look for residues of things, including antibiotics. They do these kinds of tests to verify whether or not the meat is suitable for human consumption, because there should not be any antibiotics in the meat by the time it reaches the consumer.
Would we potentially be using antibiotics in the manufacturing or development process?
Well, that will depend on what the FDA and the USDA advise, and if these companies choose to use some kind of antimicrobial for that purpose, or an antibiotic, they will need to prove to those regulators that it's safe. In the case of the FDA, where they're interested in that, and this goes back to the joint agreement that was released last year, the FDA is going to have oversight during what we would call the proliferation stage. When these cells are acquired and are brought up and cultivated and delivered to the establishment, anything up to that point has FDA oversight. And after that, once the product has transitioned into what would appear to be meat or seafood or poultry, then the USDA has some oversight for that. And whatever intervention an establishment has will be reviewed by one or both of those regulators. So they will need to prove that it works and that it's safe and that there's nothing that gets passed on to the consumer. Do I foresee there being a way of doing it without antibiotics? I would like to see them be able to do that, because I don't know if it can be done economically if they're using antibiotics at full scale all the time.
Could we potentially use human blood to replace FBS?
That seems to me a bit wild. It's an interesting thought, because there's a lot of tissue culture research that does rely on human serum, and it's possible that we could rely on it. I don't expect we will, because it introduces the possibility of spreading human pathogens into our food supply. And it just comes down to, we'll say, for example, if somebody has come down with hepatitis A and they may not know about it, and if this serum was collected from them, it would need to go through testing, it would need to go through all this kinds of verification. No, I don't foresee that would be viable, because all the things you would have to do to make it safe enough to actually be appropriate for use in food supply would be prohibitively expensive. And I'm not sure there are many people who could be convinced to buy a product that was grown with human serum. I don't know. Maybe there is a market out there for that. I don't know.
Cool. And then last audience question that we'll take on: does clean meat contain vitamin B12?
We would need to look at a sample and test it to say for sure. There's a reasonable probability that it could. After all, what's being created are the same tissues that we would expect to find in the representative animal. So if we're talking about beef, and beef is a source of B12, and we're looking at cell cultured beef, I would expect to see it there. I would be surprised if it wasn't.
We covered a little bit about antibiotics. We covered quite a bit about what these bioreactors could look like. On a personal level, what are you most interested in when it comes to seeing this future of food in combination with the cellular agriculture technology?
Okay, I like sushi, and I wouldn't say it's definitely in my top five favorite foods. I'd say I'd put it up there. And
What kind of sushi?
I really like sashimi, and I like those crunchy rolls. Those are two that really stick out in my mind. There's this little place in Austin, Texas, that does all you can eat sushi, and I haven't been there in years, but I went there with my friends once and just had an amazing time. And I ordered a lot of sashimi that night, and yeah, it was good stuff. Um, there's something they had on that menu that I guess they covered it with like a little bit of lemongrass or lemon juice. I'm not sure, but it complemented the flavor of the seafood very well. So I'm excited for the possibility of there being cell based seafood, cell based sushi that's much safer and interesting to eat. I've never tried fugu. I've read about the risks.
That's the blowfish, right?
Yeah, yeah. And every once in a while, an improperly prepared piece may contain tetrodotoxin. And so it's very risky for that. And I think it'd be really cool to be able to try a pufferfish that didn't have it. And of course, I'm talking about like the real superficial reasons. When it comes to seafood, there's other big ones like the ways that cell cultured seafood can really relieve stress on fish populations, especially vulnerable fish populations. Like, what will happen to the population of Chilean sea bass when a cell cultured version of sea bass comes on the market? Will people pay a premium for that over what they were paying before, and will that species be able to recover? That's another interesting appeal. As far as sustainability, because that's another thing that gets brought up, is whether or not it's sustainable to do so, and I'm not sure about that. I recently completed a class on food security, and we looked in detail at the sustainable development goals that were developed by the FAO. And there's this problem. Well, I wouldn't say problem. There's this challenge on us to use the same land and water that we've always had or less, and use it to go from feeding 7 billion people to 9 billion people by approximately 2050. And I really hope that cell cultured meat, poultry, and seafood are a way that can allow us to address issues of hunger, not just for keeping developed nations fed, but also giving underdeveloped nations an opportunity to provide their people with protein from a source that they didn't have before. Protein malnourishment is still prevalent in a lot of countries, and when it's combined with other factors like lack of access to sanitary water, for instance, it can be devastating. And just being able to get access to a small amount of protein can make a huge difference.
I would be totally in for an all you can eat cell cultured sushi bar.
Where it's growing as you order it.
Yeah. Wow. So back to your research, what kind of insights could your research unfold, and who, in terms of kind of category of either company or person or even government, who would benefit the most?
Okay. So with the data that we're wanting to create right now, it's it's all about detection. And where we can detect foodborne pathogens, we can save companies money by protecting them from having to do a recall. Because obviously if there is a contaminated batch, we'll say, like let's go back to the talk about proliferation in tanks and all that. So let's say you do have several different tanks that are processed independently of each other. They're in relatively small portions, but for whatever reason, one tank didn't get cleaned up properly. Some bacteria grew, but they didn't quite grow to a level where it was visibly obvious, and then that got spread further down the line, where you combined these batches into one larger batch. What I want to do is create data that could be used to prevent that kind of event from happening. Because if it's a 1 liter unit that was contaminated and we can remove that and we can save the other 999 liters from the 1,000 liter batch, that's huge. As they say, an ounce of prevention is better than a pound of cure. What I'm researching is what are the strategies we can learn to prevent contamination and minimize risk before it happens, because that'll be crucial for startups. It'll be beneficial for them because they'll be able to see and predict where and when and how to implement safety controls. They'll be able to anticipate what they should do if they do have a contamination event. And not only that, but we'll look at it further down the line when it's coming into actual mechanical processing and it's being treated like traditional products are being handled. And there's the dissemination of information on that end. And all these establishments, all these producers will need to prove that their methods for packaging, storing, and delivering food to the market shelves are safe. And they'll want to establish quality dates, sell-by dates, things of that nature. And my research will benefit them. It'll benefit regulators. And we're looking at all this evidence based. So we'll be able to find solutions that make regulators happy, make producers happy, and can reassure consumers.
So we are in the middle of a pandemic now. How has the COVID-19 pandemic affected the state of research and your lab specifically?
Well, I recall back in late February, early March, when this pandemic really got on everybody's radar. It had already been in the US, I think, for a month or two by then. And our school was quick to come up with a plan. They announced that we weren't coming back after spring break. I didn't have much time to put up my projects, but the thing about Lubbock, Texas, is we weren't a hotspot right away, but then didn't have any testing happening, so who knew what was going on? The actions that our university took pretty much froze all laboratory work for me. I've only gone to the Pathogen Lab to check on things every once in a while to make sure our equipment is intact. I am the Pathogen Lab manager, so if anything happens in that lab, it is my responsibility. Otherwise, yeah, I'm working straight from home.
And I know that a lab is probably one of the, especially a pathogen lab, will probably be one of the actually the most sterile environments you could probably be, right?
Yeah.
You can get in touch with Sam on LinkedIn and learn more about New Harvest at www.new-harvest.org. Sam, thank you so much for being with us today on the Cultured Meat and Future Food Show.
Thank you, Alex.
This is your host, Alex, and we look forward to being with you on our next episode.